So I arrived at NIH about 30 years ago for a year or two, and I never left. So I'm going to talk about influenza virus antigenic drift, a topic I actually began in my PhD in the late 70s. Here's a subtitle, Making Immunodominance Great Again. You can actually have these hats made for about 15 bucks. Yeah, well, we almost did it. I went to the website, I almost hit the button. We really should make them, be funny. It's great to work for Genius, I can just tell you, working in the US government. So here's a really pretty diagram of a flu virus, a computer-generated image from cryo-EM. HA is the most predominant glycoprotein on the virion surface. The role of the HA is to attach the virus to the host cell and initiate the infection. The virus is endocytosed into an endosome. pH is lowered. HA triggers a fusion event between the viral and the host membrane, and that's how the infectious cycle begins. The HA is really the only target for classical neutralizing antibodies, right? So antibodies that bind HA will block attachment. They can also block fusion. It turns out that neutralization in vivo, although this was predictable, because nothing is ever as simple as it seems, is way more complicated than that. And thanks to the effort of a number of labs, not including mine actually, we're understanding in vivo that many, even of the classical antibodies that I helped define, my PhD was making and characterizing monoclonal antibodies to HA, standard ones. It turns out probably even many of these in vivo neutralized by different mechanisms, including the involvement of Fc receptor-bearing cells. So here's something that we have been studying, humanity, since basically the 1930s when flu was isolated. People knew right away. There's actually a paper preceding the isolation of a human flu from Shope, he of Shope papillomavirus, who had the first isolated influenza virus in pigs. Shope immediately showed that antibodies could block infectivity. Right? Back then, antibodies were principles. They weren't proteins yet. But you can transfer them in serum, and it turns out we're still trying to figure out how these antibodies work. And now there are completely new categories of antibodies against the stem, antibodies that everybody ignored for years against the neuraminidase that probably clinically are very important, that we've underestimated how important they might be in vaccines. We just have to rethink everything about flu. Okay? And I encourage you young people to think about coming to flu. As Stacy said, there's going to be a lot of money spent on flu now because it's no longer a dirty little secret that the vaccine sucks, right? It really sucks. And in people my age, it probably doesn't do anything, best case scenario. And even in the best of years for the people who do best, which are kids, and it's really important that children get vaccinated, we're still talking maybe 60 or 70% efficacy in the best of years, right? And that's what we measure, and then as Stacy said, I mean, there are so many people infected who don't know it, and so what do we really know about flu? Not much. Yeah, not much when you get down to it. Okay, so what is antigenic drift? Drift is almost certainly the result of classical neutralizing antibodies. The virus can escape by amino acid substitutions based on single point mutations that result in antigenic changes in the HA. And one of the key questions that I started to address during my PhD in the late 70s that has never been answered is why does flu drift in people, and not in animals so much, very slowly actually in birds, if at all, and other RNA viruses like para-influenza viruses, which in vitro will drift exactly at the same rate, but in vivo in human beings, they don't really drift. We have vaccines against paramyxoviruses, they work really well. Why is that? What is it about flu? I'm not going to answer that. We do not know. This central question. You know, so it's easy as a student. You look at the textbooks, and many of them are so big now, they aren't even textbooks anymore. You buy a book so they can sell it to you, but then they give you a CD that has a lot of information, right? And it would be easy to have the impression, reading these tomes and carrying them around in your backpack, that we know a lot. And we do, compared to a chimpanzee or a gorilla or humanity even 15 years ago. But the amount we could learn, of course, is infinite. And the amount of practical knowledge we need is whole something else. What do you actually need to fix something, right? So Jenner didn't need anything, basically. He didn't even know what a virus was, much less a protein or nucleic acid. And he basically made a vaccine that eradicated the single largest pathogen of humanity, which is smallpox, still is. The only virus that is easily transmissible and highly pathogenic smallpox. So knowing nothing, Jenner solved that problem. What's left are things that we haven't solved the problem with what we know. And the point being, there's lots of basic questions for you guys we still need you to answer, right? Both for pragmatic ends and because, God damn it, they're interesting, right? They're interesting. And science should be fun. You should be able to pursue things that you are interested in, right? And you have to write bullshit on the grants, we're going to cure this or that, but never lose sight of the real goal of science, which is to have fun, right? And entertain yourself intellectually, right? That was my goal anyway. And I was able to do that. And this is just showing you, Stacy already told you this, but you've got antigenic shifts where you have a reassortment event, you bring a gene in from an avian or a swine virus, and this infects a lot of people. It doesn't actually kill any more people, except for the 1918. And if you were to add up post-1918, what has killed or caused the most hospitalizations, it is drifted viruses. And it's probably greater than 90%, because there's only been 4 or so antigenic shifts that have occurred since 1918. So drift is really important, right? The lack of having a seasonal vaccine is a very significant medical event that we need to fix. Okay, so putting some numbers on it. Antigenic drift, 100 sons of virus. So how many mutations does it take to completely, quote unquote, because nothing's ever complete, but to grossly change the antigenicity of the virus? This is a paper we published in Cell Host and Microbe. Basically, it was work that I had started in 1978 that I never published. Sat in my notebook for 30 some odd years, and finally we got back to it. So advice for young scientists, never forget your old data that you haven't published, right? You can always go back to it if you're still in the field and get good papers out of it. There's like 5 papers from my lab in the last 10 years where the first experiment of the paper I did as a graduate student, right? So it's, don't throw out your notebooks, right? Keep remembering this stuff you don't know. So what did I do? I took monoclonal antibodies. I worked with a guy named Walter Gerhard who made the first monoclonals to flu. We had hundreds of monoclonals. I asked a simple question: how many mutations does it take to abrogate the binding of all of these hundreds of monoclonal antibodies? It turned out 12 selection events, more or less, were what required, okay? So 12 independent events changed antigenicity. What is the frequency of a spontaneous escape mutant with 12 residues? It's about 12 to the minus 50th. Okay, my first paper as a PhD student was showing the escape frequency of single mutations is about 10 to the minus 5th, right? A little higher than that. Okay, well, let's put some physicality to that. 10 to the 50 virions. Virions do not weigh much, but 10 to the 50 is a big number. That's 10 to the 32nd kilograms, right? Or 10 to the 35th grams. The Earth weighs 10 to the 26 kilograms, and the Sun weighs 10 to the 30th kilograms. So we're talking about 100 sons of virus to get 12 mutations if it was going to be spontaneous. So obviously that is not how drift happens. And I've set this whole thing up, you knew that anyway, but it's still a fun number, right? It's always fun when you get into logarithms. Okay, so how does it really work? Well, the way it really works, and we know this from isolating viruses from people and animals, you get viruses out of some animals with 1 or 2 mutations, and that really can abrogate antigenicity, functional antigenicity in neutralization assays. Okay? So what that points to is the importance of immunodominance. Immunodominance, right? That we're going to select certain proteins or certain regions of proteins and focus our immune response on that. What's known about immunodominance in adaptive immunity? So ID, the phenomenon of unequal immunogenicity among antigens in a complex immunogen. What's known about it for T cells? Fair amount, actually. Jack Bennick and I spent most of our careers at NIH, 30 years, trying to establish some rules for immunodominance in CD8 T cell responses, and we don't know everything, right? But we found out quite a bit. So there are pretty good rules for that. For B cells, surprisingly, because antibodies were discovered way before T cells, and people have been studying them for, basically since the 1930s or earlier, there is very, very little known about immunodominance in B cell responses. Again, you'd think, there are immunology textbooks, you'd think we know this 50 years ago, but we don't, actually. And what work there is limited to either incredibly simple systems like sugars, haptens, right? Or some bacterial systems. But even there, not a lot of great immunology. So this is flu, cartoon. It's a relatively complex immunogen, at least compared to something like ovalbumin, with multiple targets for antibodies, right? The virus has, this is basically the structural proteins have. Some are ironically named because we thought they were non-structural. Always be very careful when you use that word, because you can only be wrong. You can only be right one way, and you can be wrong in an infinite number of ways. So it turns out the non-structural proteins are some in the virion. There are 4 major influenza virions, however. And Meghan, who just left my lab, she was with me for 6 years, outstanding postdoc. She's actually looking for a job. Brilliant young influenza virus researcher. I hope to get back to her at the end of the talk. Meghan asked a very simple question. What is the antibody immunodominance hierarchy among flu antigens? In other words, what are the antibodies predominantly raised against? So how do we do this? Immunize animals with an activated flu virus. We can do it with live, but you'll see why we use inactivated. Just do something really simple. Just test the sera in an ELISA. You get a titer. Okay, well, what's a titer? Well, that's a problem, a titer. That's a problem with measuring antibodies, because counting them is not easy, because counting them is based on their concentration and their affinity. And we all throw around the word specificity as if it's a simple term. It is not. Every antibody binds every antigen at some affinity. Okay, and that is really tricky, because what is a relevant affinity? Again, another low-hanging fruit. We don't know what a relevant affinity is for an antibody. I can tell you in ELISA, if the affinity is less than 10 to the minus 7th molar, you will not detect it, right? Because it'll fall off during the washing steps. But does that mean it can't provide protection in vivo? No. What literature is out there? There is a few papers. A guy named Nigel Dimmock studied this. There was basically no correlation in protection and affinity in antibodies that were basically pretty high affinity. So there's another hole in our knowledge, this relationship between affinity, or actually avidity, because antibodies are multivalent and so is a target, and their biological activity. Okay, so we have a titer to measure, and we can just use ELISA against various forms of antigen, and then we're going to determine the dominance by titer, and if you're interested, this was published in eLife a couple of years ago. And this is a bit surprising. So we're going to normalize according to abundance, and what's the most immunogenic protein in flu? It's neuraminidase. Whoa. I would have guessed hemagglutinin, and I think everybody else too. But in terms of abundance, Na is not a very abundant protein, and yet it's highly immunogenic. And you say, well, if Na is so important, it should drift. Guess what? It drifts faster than HA, right, if you look at sequence. And where does it drift? It's in the globular domain where the antibodies bind. So does this mean that it's being selected? Almost certainly. So why have people ignored it for 60 years? Because we're idiots? And I put my hand in there too, probably. Okay, so you can see the immunogenicity. Na is most, HA, NP a bit, and M1, which is the most abundant virion protein, and this has been known for a long, long time. This guy is basically not immunogenic, right? Not immunogenic. What the hell? Okay, that was done with mice, but we all know that mice are ridiculous, and mouse immunologists should be crucified because they waste their time studying something that is completely irrelevant for human beings. I hope you hear the sarcasm dripping from those words, okay? Mice lie, right? Obviously. Cats will build a wall. You can see who my favorite politician is. Okay, so what Meghan did was ask another very simple question. Again, you would have thought this was known. Are there universal rules for immunogenicity? If we could find a completely different humoral immune system in the universe, right? How would they respond to something like inactivated flu? Because the odds are flu wouldn't be able to infect this thing. So we didn't actually have to send Meghan to Mars to find Marvin. We could send her to Atlanta, where she found the guys who work on lamprey, right? So Max Cooper and colleagues made this amazing discovery 10 years ago. Max, as a student, again, never forget what you do as a student. He wasn't quite a student. He was a young MD fellow doing research in Bob Good's lab, very famous lab at the time in the 1960s. They discovered that things like lamprey, right, jawless vertebrates, they make antibodies, you know, just functional. You could stick red blood cells in them, take out the serum, they will agglutinate the red blood cells. That's what they discovered and published in JAM. And Max never forgot that. eventually got back to it, cloned what the relevant genes were, and guess what? They're not immunoglobulins. They are completely different from immunoglobulins. There are two crystal structures solved by Ian Wilson's lab showing the interaction between what the lampreys have, which is called a VLR, with the same antigen, which is lysozyme, and a typical immunoglobulin. Okay, the structure is completely different. The genetics are incredibly cool. This, to me, is a Nobel Prize. I think this is amazingly cool science. Okay, so in collaboration with a guy named Brent Heron, who Max trained but had his own lab when we did this with Brent. Wait for the curtains to open here. Meghan immunized lamprey, or Brent did, and sent us a serum. And we can ask the question, and forget about the details, it's in eLife. What is the immunodominance hierarchy in terms of the titers? Now, titers, HA, NA, NP, M1 very low. That's exactly what's true in lamprey. Exactly what's true. So send Meghan to Mars, you see exactly the same thing. Okay, and then we said, okay, well, sure, maybe they recognize the HA, but they can't recognize the same regions of HA, at least that's what I thought. But in fact, they do. And the way we did this was taking escape mutants, again, that sequential 12 panel I told you about, where we selected the escape mutants with mouse monoclonal antibodies, and we asked, did the lamprey antibodies care about that? And they did, in the exact same way, basically, the mice did. Which means the lamprey not only recognize the same part of the HA, they're affected by the same mutations. To me, that was a holy shit moment, right? I did not expect that. I don't think any of us did. Okay, so what we found, and this I didn't tell you, is that the head is immunodominant. You make a really, really crappy response to the stem, and everyone would like to make a good response there because it turns out they can protect mice and maybe people, and that's where a lot of the effort for universal vaccination is going. But the stem, when it's attached to the head, is a poor immunogen. Okay, and what Meghan found was that mammalian, I didn't show you this, but bird, lamprey I showed, recognize the same antigenic sites in a similar manner. We actually have a really nice collaboration with a guy in Baltimore named Marty Flanick, who's the world's expert on shark antibody responses. We actually have immunized sharks with flu. Some of these are 12 feet long sharks that Marty has in tanks swimming around in Baltimore near the aquarium. And they respond the same way as every other vertebrate, basically, even though they have a really cool special species in shark called Ignar, single chain. Okay? This did not establish the dominance hierarchy of the globular domain, however, right? Which is really important because that's how I started, right? We're driving drift by making monoclonal responses to individual sites. So to study that, we have to do something else, right? We have to come up with a new strategy. So here was our strategy. We designed 5 new HA antigens, in this case HA viruses, that we call Delta 4, right, to enable the response, antibody response at the level of polyclonal antibodies to each of the sites. And the way we do that is by taking mixtures of antibodies against one site, selecting, doing it again, selecting, doing it again, selecting, until we knock out 4 sites, leaving 1. Right? And we have to do that 5 times. This took us a couple of years. Because you're running into dead ends, you get a mutation, the virus isn't happy, you got to start again. Okay, but we eventually could do this. And then we could test the serum by ELISA to measure site-specific responses, or we can actually count B cells, which is really important. We can count B cells by using HA probes, recombinant HAs, produced by our collaborators that we can use for flow cytometry. And eventually we wound up with a 7-virus panel. Wild-type virus, our 5 Delta 4 viruses, and then sequential 12, which is missing all of the globular antigenicity. And this is Davide Angeletti, who has been with me for 5 years, is choosing now among labs to start his own lab in Europe, a really fantastic postdoc. This was published in Nature Immunology last year, and Davide could ask these questions. Is there immunodominance hierarchy among the 5 sites? If so, how is it affected by form of virus we give, form of immunogen, route of infection, genetic background, pre-existing immunity? You can read these, right? You can ask all these questions. And one of the keys is this new technology, new because it should have worked before, but people have gotten so much better at making recombinant proteins now that stuff you tried before that kind of worked a little bit now really works well. So this is an HA that the guys at the VRC, who we collaborate closely with, figured out how to get rid of the sialic acid binding activity by mutating a single residue in the sialic acid binding site. It's got biotin at the C-terminus, and so you can just stain cells with this now and count how many B cells are specific for your antigen. This is really important because we get around this whole problem of titers. And I'll show you we can even get around the problem of avidity. And what Davide found was that the specificities of germinal center B cells temporally evolve. So you start with a response against this site called CB, which actually we knew from Walter's work back in the late 70s, early 80s, that this was immunodominant very early. This does come up, but with time after infection, this is infection of lungs of mice. By day 21, we start making a response to SA. By day 28, 4 weeks after infection, we basically now have a very diverse response. This is not how T cells work. T cells, you make a response against X number of peptides, which keeps growing as the sensitivity grows, and it changes a little bit over time, but not much. B cells are different. So what's going on now in the germinal centers is really interesting, because what happens is B cells enter the germinal center, they start somatically diversifying, they start competing with themselves, but obviously the competition is not so severe that we can't come up with completely new specificities. And I'm not telling you these are being somatically diversified, these are actually precursors that somehow fight through the early competition where we can't even see them to make it weeks later. Okay, so the germinal center must be constructed in such a way to enable this kind of competition, okay, where competition doesn't kill everything. And if we measure serum antibodies, it's not perfectly parallel, but it's very close. So the germinal center is a good measure of what eventually is going to show up in the serum, and that's good for two reasons. One, germinal centers are pretty easy to measure, the germinal center B cells, and two, that's reassuring because you don't really know where the antibodies are coming from. Right? Even a mouse is a relatively large place, and there are B cells in all sorts of nooks and crannies. And showing this correlation is important because it implies that the germinal center is the primary source of the B cells, which are probably going to be plasma cells and plasmablasts that are making the serum antibodies. Okay, this is cool. This is a paper we had previously published in EmBio, Greg, Frank, and Davide. What we showed is something that you might predict could work, although the referees disagreed with that in this paper, which is why it's in EmBio. And what we showed was that if we just, as we stain the cells in this assay for germinal center B cells, if we just dilute the HA and make a plot like an affinity plot, it actually works, right? It kind of shouldn't by the law of mass action, but it does. So the amount of HA required to stain half of the cells basically predicts the affinity of the antibodies that those B cells would make. So I think this is really important because you can actually measure the affinity of the B cells at the population level anyway by this very, very simple measure. And the referees for immunology journals told us this could not possibly work. And we told them it does work, and that wasn't enough. Right? We took a hybridoma where we knew the affinity, showed it worked, but I don't know. You know, people say they don't believe something. That's always a problem, right? You should not be allowed to write, I don't believe something. You can, you know, unless you're a priest, then you can say that, right? But if you're a scientist, you can't say that, I don't believe it. You can tell me why I think it's bullshit, but you can't tell me I don't believe this, okay? So that was quite cool. And what did we find? Nothing super interesting. I think it's a really useful assay, but basically found within the margin of error actually, that there aren't great affinity differences between the antigenic sites. Even though the CB starts a lot earlier and you might think it would get a lot higher, it doesn't. Right? They're all about the same within a pretty narrow range. And this is something quite cool that we can now do for the first time, is measure the efficacy, the biological efficiency of antibodies against different antigenic sites. Right? So we kind of knew from the monoclonals that this microneutralized stuff was true, that the SA and SB sites were better than the other sites in terms of the efficiency of antibodies per amount bound in terms of their function. It makes sense. The SA and the SB sites are at the top of the molecule near the sialic acid binding site. This is microneutralized in vitro. I'm not talking in vivo. And then this was a real shock. So this is this incredibly cool and beautiful and historic assay that's kind of useless, hemagglutinating inhibition. But those of us who trained in flu love this assay because it's fast and you get results. But this is really amazing. SB and monoclonal antibodies are the most efficient at giving HI in vitro, right? Most efficient. With polyclonal sera, they're the least efficient. And we cannot figure this out. I have tried. We spent many, many hours doing all sorts of reconstruction experiments. We have not figured this out. Why should you be interested? You should be interested, and this is a little arcana from flu, obviously. Why you should be interested is that it points out a very important point. If you want to understand antibody responses, monoclonals are nice. They can inform you what might be true. But at the end of the day, you have to understand how polyclonal antibodies work together. And this is complicated because not only can they compete with each other, they actually can enhance each other's binding. That is not uncommon. take antibodies against one of the antigenic sites, they very often increase the affinity against another site, probably by limiting the conformational gyrations to make the on rate higher. Okay? However it works, this is really important. And this is a really important discrepancy that points out the importance of working at the level of polyclonal antibodies. Okay, so you'd like to know what antibodies you get depending on what the vaccine is? And what we can show here, what Davide can show, is that it varies. So that's really important. What's one of the vaccines? What's the typical vaccine? Is you give the antigen IM. Why would you ever give an antigen IM if you want an immune response? Good question, actually, right? We all do it. It works. But what the hell is the muscle in terms of the immune system, right? So it isn't terribly surprising that you get a different specificity in your response if you give it IM versus IN, intranasally, a real infection, right? And these important differences have not ever been studied at the level of human beings, right? Never been studied. Something else actually predicted by a friend of mine, Rustam Antia at Emory, was that antibodies should suppress the response to the same antigenic site in vivo, and they actually do. Okay, so if you make an antibody response one way or another, say to the SB site, and then you challenge with virus, you actually have a decreased response to that site. So that kind of simple competition works. And in fact, what's remarkable is that the competition in vivo is broader. The SA antibodies will block CA and SB, but in vivo, the effect is selective, which I think is really interesting. Not sure what that means, but it's really interesting. That doesn't match. Okay? But that's something else that is sort of the opposite of what we think is the problem of original antigenic sin, where if you make a response to something, you'll get a better response and it'll dominate everything else. This is sort of the opposite under these circumstances. Okay, so what did Davide find? As with anti-flu, CD8 T cells, B cells generate a predictable dominance hierarchy specific for globular domain antigenic sites. Antibodies to the globular domain sites have different functional activities but similar avidities. The hierarchy varies with route of immunization. I didn't show you this, but mouse strain. We're now mapping this with the guys at UNC using collaborative cross mice genetically. Pre-existing antibodies suppress response, so we discovered OAS2, original antigenic suppression. What do I have? 2 minutes? 3 minutes. Okay, so I'll quickly go over this. More of Meghan's work. Elephant in the room with flu is what the hell are the glycans doing to antibody responses? This is leave through the gift shop. Exit through the gift shop, a really funny movie about art. Okay, so heme gluten is glycosylated co-translationally. Stem glycans are highly conserved, essential for biogenesis. The head is variably glycosylated, right? And what controls the size of these glycans, because they get processed, nobody knows. Really, this glycobiology, even though people have been studying it for a while, still in its infancy. So many unknown questions. What do they do? Lots of things to the HA. They influence its receptor binding, but something they absolutely do is they block antibody binding. And Stacy told you one of the problems with the vaccine is that perversely the virus in the last few years has decided to put sugars in the most important antigenic sites. And I think this is the main reason why the vaccines for the last couple of years have sucked, because of that very function. And they do other things. So Meghan asked a very simple question. Is there a pattern to the glycosylation? And you can do this in a couple of ways. You can predict where the glycans are. That's what everybody does. Or you can go old school and measure the number of glycans just by STS page. Okay, so this was a lot of work, not in the STS page. This is quite simple, but in purifying all these viruses. Meghan made about 100 viruses, purified them, did it a bunch of different times, measured the mobility, and came up with some interesting things, right, that weren't basically known. So this is just plotting a number of predicted glycans versus molecular weight, and she found some simple rules. That the H1HA, right, adds about 4 kilodaltons per glycan. The H3HA puts on a smaller glycan. Okay? Why that is? I don't think anybody knows. I don't know. I don't think any of the glycobiologists know. But the HA does that, and these viruses are very good probes then for figuring out what the hell the glycan modifying enzymes are doing, if you're a cell biologist. Something else Meghan found is that by the end of the evolution of these viruses, they could add about a total of 21 kilodaltons to the head and still function. Okay, now that's not an absolute limit, because Palazzi has shown you can put a lot more on there, but this never happens in nature. which tells you something important about the capacity of the HA to add glycans, which is almost certainly why HA is not like GP160 and hides from the immune system just by covering itself in sugars. It would like to do that, it just can't do it and still function. Okay, and what Meghan found, I'll run through this very quickly. This is not published officially yet. I mean, not in a journal, but it's on this bioarchive server, which I am a huge fan of. I think this is a great thing for science. Meghan found that for H1 viruses, glycans are initially added every 5 years, then they reach a limit. Okay, and after the H1 limit is reached, basically every 10 years or so, they swap glycans between sites in the head. Okay, so going to H3s, it's on a slightly longer clock, a 6-year clock, until 7 glycans are added, and so she came up with these laws for glycosylation, some of which I just told you, and this I'll get to the end. Okay, so what predictions could Meghan make? She actually predicted the addition of a glycan to the virus in 2015 that happened. And if we're going to make more predictions, especially about the future, this is difficult, but Meghan can predict this. I'm going to end right now. That the pandemic virus will add another glycan in 2021. You can make a guess where it is. She would also say that H3N2 can't add anymore, and it is ripe for replacement by a new pandemic virus. Right? It's eventually going to happen, and based on the glycans, we would guess pretty soon. I mentioned everyone who did the relevant work, except for the guys at the VRC. Adrian McDermott's lab provided the HA probes for us. James Stevens also provided probes from CDC. Brent Heron was a guy at Emory. Didn't get to what Martha contributed in the talk. And I just want to mention, I have an EBook for young scientists where I give you practical advice on how to succeed in science. You can get it on Amazon for 99 cents, which is the lowest price you can set for a book. But if you email me, I will send you a link to a Google file, a Google folder, and you can just download the book. Okay? And if you do that, please give it to all your friends, anyone who wants it. And I'll just end by telling you I'm in the Laboratory of Viral Diseases at NIAID. This is a lab of 8 PIs. It's a fantastic place for virology, for viral immunology. I have openings in my lab. Many of the other guys do too. If you're interested, write to me, and I can sort you out in terms of the other labs. And please take a look at our webpage for what everyone's doing. It's a great group of people.
Okay, questions for the speakers?
So 2 of you mentioned original antigenic sin, and I think Stacy, something to like that's the most important, was very critical for how you respond to future vaccines, I guess it was what you were suggesting, I'm not sure. And then Jonathan mentioned it as well, but in a different manner. So could you maybe discuss that among yourselves and explain the relevance for vaccine development and just basic immunology?
Well, I definitely think your immune history is important in how you're going to respond to a primary infection or a vaccination. And it's something that none of our current animal models address at all, right?
Well, there's a lot. Joshie Jacobs at Emory has got some really beautiful papers on OAS. So OAS is a phenomenon where it's basically immunodomination by the first antigen you've seen. And there's a famous paper from Rob Webster and Fasicus St. Groth, Disquisitions on Antigenic Sin from 1960 that I urge all the students to go back and read. Fantastic. But Joshie showed that the outcome depends on so many factors, not the least of which is adjuvant. And so OAS, easy to document, also clear it's not, in terms of the antibodies, it's not like you can't get new antibodies, you do. But here's the thing, is that what we measure in vitro in terms of our NUT is not correlating with protection in human beings, right? And that, in this case, I will admit mice, they don't lie, but you can never recapitulate flu in any animal model because if you're 15 years old, you've been exposed 15 times to flu. And how are you ever going to figure that out in an animal? And for most viruses, it doesn't matter because they're just protected from the beginning, but for flu, it matters. And so modeling this thing is a bear.
Yeah.
It's a bear, Dan, and it's a severe problem. And we don't have you can't believe any of the in vitro correlates for what is protecting a human being.
And honestly, when we go to WHO twice a year to decide on what flu strain should be in the vaccine, we don't even discuss the impact of immune history or immunodominance when we're making those decisions. It's all about the viral sequence and the results from hemagglutination inhibition assays. Because how would you even get at that when making decisions about the vaccine?
But it's at the center now of influenza vaccinology, and people are starting to ask the question, okay, we have a baby. What's the best thing to do? Because we may imprint this baby for the rest of his life or her life. So should we vaccinate him? And if so, do we want to give them the live attenuated vaccine? We'd probably say yes.
Yep.
Would it be better to let them be exposed to real flu? It's probably not that risky. And if you had much better lifelong immunity, would this be preferable? And the problem is to make a rationally informed decision, the data aren't there.
Yeah.
But people are now really worried about this, and which is good, because we're finally really squarely addressed the issue. And the Institute now, Tony Fauci, the head of our institute, is 100% behind in fixing seasonal vaccination. So there are going to be a lot of resources there, and there's no guarantees here. Probably it's 50-50. We had a meeting recently. People were asked to vote, what's the universal vaccine going to be? 50-50 T cells versus B cells. I don't want to give people the wrong impression. T cells are still in the mix as well. And if you really, I think, queried the field, what percentage of people believed a universal vaccine would really work universally? Probably 0%.
Yeah. Yeah. Honestly.
Yeah.
Yeah.
I had a question for Jonathan.
Neela.
I'm really struck by your result of the antibody specificities between mice and lampreys and sharks and so on. And as you said, one shouldn't forget old results. So I remember when I was a graduate student, I was working on antibody specificities to lysozyme, which is a soluble protein. And the antibodies would recognize multiple epitopes all around the protein. So it wasn't just a fixed, immunodominant epitope that was there. So I was wondering if the result that you had anything to do with the fact that HA was part of the virus rather than a soluble protein.
Excellent point. So we didn't test that directly, but in mice, it doesn't matter much. We can give HA as just basically a free protein, although it may be micelles. We don't think it is, but it may be. But we basically get the same dominance hierarchy, more or less.
So what do you think is the reason for that?
So the fine specificity anyway, there will be differences, right, between the dominance of the various sites. What's more striking from the lamprey data is that the same hierarchy of proteins, right, that are in the mixture, and on top of that the head always beats the stem, right? And you can make if you just have a stem, and it's not easy to make native stem, but people have figured out how to do it from the protein engineering standpoint. If you make native stem, it is incredibly immunogenic, right? And so the key with the stem, and we haven't published this yet, but Davide has found, it's the physical association with the head. And we think there may be a very simple explanation that the intrinsic avidity of the B cells for the head is tenfold higher, and that seems to be enough to kill the stem immunogenicity in a primary, which is interesting, right? But what I think the most important implications of our various species comparison is that the immunochemical properties of a protein govern its immunogenicity, right? And just somehow it's got to do with protein-protein interaction, and whether you're an IG or a VLR, the same rules apply.
I had another question for Stacy. In your obesity studies and the correlation with viremium, do you think the adipocytes or the adipose tissues play a role?
That's a fantastic question, and we are just starting to look at that. Everything we've done, we've focused at looking at the lung per se, and while the lung is not obese, there's plenty of adipose tissue surrounding the lung in these animals, and so we're starting to look at how does the adipose tissue itself actually impact the virus, and we know it's going to. So those studies are just beginning.
Can you do a fat transplant?
So we've talked about doing liposuction, so a little bit the opposite to see if we could actually protect the mice. But we haven't tried
cool.
doing,
Yeah.
a fat transplant, but that's a really interesting question. I can tell you that even when we make these primary cells, whether it be from nasal or lung, from lean versus obese animals, they behave very differently in terms of what they're releasing and viral titers. So,
What's it called when you attach the mice to each other?
yeah, I know, because we've had more people say when you sew them together, basically.
Yeah, that would be cool.
Yeah.
Right.
I don't know, even though I'm chair of the IOCOOK, I don't know if our IOCOOK will actually let us do that.
But what guys at Minnesota do that?
Yeah, and so that would be an interesting thing to do.
Dave Massapus knows how to do it.
Okay, that'd be cool.
Stacy, what happens when you put your mouse on a treadmill and make them lean again?
So we want to make everything fat, not skinny. But there have been actually a few studies where they've put mice on diets or in something we're going to look at for vaccine responses, but it will impact the immune response to flu. So that's one thing people have asked us is if we take these diet-induced animals and we actually have them go back to being lean, does their body still think they're obese in terms of their responses to flu? And we don't know the answer to that. But one study that we've done that hints that it's not obesity per se, but probably the metabolic state, is that we have mice that are genetically obese, but they better control glucose metabolism, and they don't have as severe of the disease phenotype, as well as they produce better vaccine responses.
I had a question for, a couple of questions, but maybe I'll start with one for Andy, which was, you can probably predict what I'm going to ask. But your reporter assay, I think, suggested that IFIT2 preferentially binds the viral mRNA, but does not boost host RNA translation. And I was wondering how you were thinking about that. You kind of hinted that it might be codon optimality.
Yeah, I mean, so it's a great question. What is IFIT2 seeing and how is it seeing it? And I'd love to have an answer for you. We have many thousands of dollars of sequencing data that told us nothing so far. But what we could tell you for flu, a couple of things that we thought might be important and have narrowed down. Flu messages are a bit unusual compared to host messages. They've got really short UTRs, and the way flu puts on a poly-A tail, it's also short relative to a host transcript. Those things aren't important at all. So it's not the UTR, it's not the poly-A tail. We can drive flu genes using a CMV promoter, just the open reading frame, and that's sufficient. Flu genes tend to have shifted GC contents, especially as it hangs out in humans. The longer it goes, the less GC you are. We think that might be part of it, that there's something special about the, if not codons per se, the general sequence composition. And the reason we think that this might be where we're going is that when IFIT2's structure was solved, they looked at specificity, and while it was all in vitro, it preferred AU-rich elements. And AU-rich elements are common in the UTRs of interferon-induced genes, specifically cytokines and so on. So we think that the real role of IFIT2, I mean, this is all speculation right now, is to help drive and boost these antiviral effector genes, and that flu has figured out what the rules are for that and has kind of slotted in. But there's a long way for us to go to actually demonstrate that.
And then maybe a question that all three of you might be tired of answering, but for this audience, I'd be curious if you could comment a little bit on the moratorium.
So moratoriums, I don't know if people know, so for the past 3 years, we were under a moratorium where we couldn't do any gain of function studies. And
Unless you got
a disposition from,
which we did.
right, which you could get from NIH, but like at St. Jude, we're very conservative, so we didn't even get to ask. It was just our institutional biosafety committee said, you're not doing anything. Gain of function included passaging viruses in mice, looking when you have new drugs, asking can they become antiviral resistant or can we get vaccine escapes. So it wasn't making H5 viruses that could transmit in ferrets. It was very basic virology was impacted, because if you think about it, if you take a virus out of a person and you put it in cell and you want to adapt it to grow in the cell, it's a gain of function. We couldn't do those studies. So we were looking at the impact of obesity on viral populations through passaging. Those studies have been halted for 3 years. And the bad thing is, in the past, 8 years, at least since I've been at St. Jude, that's our second moratorium. So the first moratorium lasted 2 years. This
They're
one was
completely
3 years.
idiotic.
Yeah.
You know,
and-
it wasn't the government. This came from outside forces, well-meaning but misguided people. Very few knew anything about what they were talking about. They were not virologists. And you can stop us from doing basically trivial experiments. I mean, if you ask flu virologists, make it more dangerous, no one would know how.
Yeah.
The birds did not read the ban. And while we're doing a mutation here or there, the virus is happily putting trillions of mutations in per year. And it's completely crazy to think you're going to do anything about that. So this was just completely idiotic. And it's going to sound melodramatic, but when you restrict research, people will die. I know I sound like Jack Nicholson here, right? But people will die because you know less.
Right.
And it's well-meaning, but in the end, it's self-defeating.
Well, and it only stopped people that are funded by the US government. So the studies continued in other parts of the world, and I think where it really hurt the flu community is the number of trainees that want to go into flu has dropped astronomically because, you know, all of my people had projects impacted by this. And they say, no way in hell am I going to do flu research if every couple of years I'm going to be under a moratorium.
I mean, the danger is zero. We were working on a virus known to not be able to infect people, the classic PRH strain. It took 6 months to get that around the ban. And it just, people, it's once, here's the problem in Washington is, it's, you know, it's just cover your ass mentality once flags get raised.
Yeah.
And Tony, I think, did a really, really good job in just absorbing the blows and then in the end prevailing, but it was a really bad episode.
Yeah. And while the moratorium's been lifted, the guidance that's provided primarily to institutional biosafety committees is pretty poor at best.
Yeah.
And so you have these IBCs that really don't understand it that are going to be making these decisions. And most are going to err on the side of caution because they don't want to end up in the newspaper.
And I would also add, it's not that there was a problem before. We weren't cowboys just
No.
doing things willy-nilly.
Yeah.
I mean, the amount of regulation and
I mean,
paperwork
Oh, yeah.
and
has there
approval.
ever been a single known example of a laboratory infection?
No, never. And all of the data that were used to generate these
Yeah.
scary scenarios
Yeah.
by a few people that were waving the flags were all based on bacterial exposures.
The numbers they had
or-
were just bullshit.
Yep.
Yeah, just complete bullshit.
But they convinced people.
They did, and again, I think they were well-meaning. I don't impugn their motives, but they just didn't know enough, and it was really bad.
And I would hear that at study section. Oh, these flu cowboys, literally. You know, they're just going to make something that's going to kill all of us. And you're like, what?
Maybe we all
we-
need hats and horses.
Yeah, I mean,
Yeah.
it's too, it is so completely unpredictable what any mutation will do, and that's the other thing. If you want to be honest about it, every single mutation you put in any virus is a gain of function virus.
Right.
You just don't know what that function this thing gained is, right? And often a loss of function, as Andy was telling us, is a gain of function. You never know. No, it's a hall of mirrors with the host and the virus. So you can basically say, okay, you can't ever do any mutations, and this would be completely stupid because you pick a plaque, you have a new virus. That's a gain of function. Pick any plaque with an RNA virus. It is a fish that's slippery. You can never pick this thing up, right? It's completely ridiculous and maddening. You'd go insane if you thought about it too much, right? And the problem was there were young scientists, tenure-track scientists, whose whole careers were based, whose research was banned.
Yep.
So I have a clinical relevant question for the scientists, probably for Jonathan, but others can comment. So the first monovalent LAIV publication from Dr. Masab was in 1969, and LAIV was licensed in 0304 as a trivalent vaccine. And so I'd like to know from the scientists what is being done at the scientific level to understand the problems. Over the last 2 years at the Centers for Disease Control had de-recommended the LAIV vaccine to be utilized, even though it's still licensed and available. And is there something with the head? Is there something with the stem? Has the Laboratory of Viral Diseases been sought out after by the Centers for Disease Control and the other people to find out what is really going on? Do they really have an expectation that the pharmaceutical industries can understand this? basic science level on the biology of these viruses. So what is the mystery here that we're missing? And the reason I'm asking this question is I did the clinical development for FluMist from 1996 to licensure in 2003, 2004. So I know a lot of history. I know the 93% efficacy in young children. I know it beat the inactivated vaccine in a worldwide study at 8,400 babies. What happened biologically that you can tell us from all your studies that can get the LAIV vaccine back on track for infants and get the CDC to re-recommend it? Because maybe LAIV is just ahead of inactivated influenza virus vaccine. 2 years of low efficacy or effectiveness, what the apparent study showed by the CDC, and now they're showing it for the inactivated vaccine. So what's going on?
Well, I think one important thing to note is that the LAIV not being used was only in the US. It's still being used around the world. In fact, UK just started using, and it's very effective in other countries. So there's been some talk of production issues.
Still the recommended vaccine in the UK, Canada, Germany, Israel, but not in the US. So what's happening in the US? Is there some epidemiological factor? Is there something, but it seems to me there's something more at the molecular level. We reassorted in the HA and the NA gene into the 6-2s, and then we went into plasma genetics and did it that way, but we still go back through eggs. So
Right.
what's really going on? Thermal lability? I mean, people are just throwing out crazy ideas.
So it wasn't all components of.
Maybe you can bring the rest of us in.
Sure. So, and you guys jump in. So there's a live attenuated influenza vaccine, FluMist, that has a cold-adapted mutation so that it replicates, it's attenuated replication, but in the upper respiratory tract. Very effective vaccine, recommended especially for children because it's easy to administer to them, but for people through 49 years of age. And 2 years ago, CDC came out and recommended, due to low efficacy, but it was only for one of those three at the time, components, that was a problem, but they recommended it no longer be used. The rest of the world is still using it. I think it's still very effective in children in other parts of the world, so we don't really understand what happened in the US that we had this low efficacy. And some of the discussions have been production issues. Once we put in the fourth component, perhaps there was viral interference that went on. So when you add another virus, that you get competition amongst the viruses for replication. So think about 4 viruses replicating in your respiratory tract, and one replicates faster than the others and sets up an interferon response. Yeah. That's going to decrease the amount of replication for the other 2 or 3 components. And we know this happens, and that could be one reason, but I don't know if anybody really is exploring it. I thought it was, I was shocked when CDC came out and recommended that it no longer be used, because it is such an important component of our very limited arsenal.
So it's 17 million doses. It's still licensed. That's
Right.
what was up to in terms of production.
But
it's not covered by insurance if it's not recommended. So nobody's going to pay $40 or $25 or any bucks to get a vaccine. They're going to just not. And is there any relationship between the last 2 years of not LAIV getting out there and impacting herd immunity with children to what we're seeing now with the outbreak in Australia and the outbreak here?
Well, I think Australia is still using LAIV.
The basic answer is we don't know.
Yeah.
And it's complicated, right? Because even something like counting the relevant number of particles is not easy. We published a paper a couple years ago now where we showed that despite the dogma that packaging was almost perfect, it's not only not perfect, it's deliberately imperfect. So 90% of virions are missing one or more gene segments. People never account for this. And I'm sure in any different biological preparation, the number of segments missing will vary enormously. And that's just one of many factors that would make things not bioequivalent, even though you think they're chemically equivalent. So it's all really, really tricky. And I think the secret is that we're not idiots who work on flu. It's just harder than all the other viruses somehow that we've worked, except for HIV. The ones left are the harder ones. right? And when things work, they work, and we don't really know how. We have one of the best words, my favorite words in medicine, is correlate of protection, because that's exactly what it is. It does not mean that something is protective, it means it correlates, which means there could be 4 things distant from that actually is the mechanism, right? And then there's not a mechanism anyway, it's always much more complicated. And I think we're just coming to grips now with the complexity of immunity. It's complicated. The live attenuated vaccine should be the best vaccine, because not only do you get a natural infection, you probably get, because it again has not been characterized, a much better T cell response to it, right? So it should be a big plus.
So I switched to norovirus 9 years ago. I'm working on that vaccine now.
Thank you. That sucks.